There are two types of cycles that electrons will go through in a plant in order to get the energy that it needs, and they're called the linear flow and the cyclic flow. The linear flow starts when light photons hit the light-harvesting PS2 complex, boosting one electron to a higher level. This electron gets passed to a nearby pigment that then gets raised, and the process continues passing from pigment to pigment until it reaches the P680 pair of chlorophyll A molecules in the PS2 reaction center complex. This then excites an electron in the pair of chlorophyll A. After the electron gets passed from the P680 to the primary electron acceptor in the chlorophyll, it is referred to as P680+ since it is missing the electron. In the chlorophyll, an enzyme catalyzes the water molecule splitting into 2 electrons, 2 hydrogen ions, and an oxygen atom, which are supplied 1-to-1 to the P680+ pair. Each electron replaces one transfer to the primary electron acceptor. The H+ are raised to the thylakoid space, and the oxygen atom joins one generated by another water splitting, making O2. All of these electrons then pass through the primary electron receptor of the PS2 to PS1 via an electron transport chain made of plastoquinone. This fall of electron provides energy synthesis of ATP, and as the H+ get pumped into the thylakoid space, it contributes to the proton gradient. The electrons in the PS1 reaction center complex get passed to the P700 pair, which creates an electron hole and transforms P700 into P700+, which can act as an electron acceptor which reaches the bottom of the electron transport chain from PS2. These photoexcited electrons get passed in a series of redox reactions from the primary acceptor of PS1 to a second electron transport chain through the ferredoxin. NADP+ reductase catalyzes transfer of electrons from ferredoxin to NADPH, whose electrons are more readily available for the Calvin cycle. The cyclic flow is very similar to the linear flow, but it uses the PS1 and not the PS2, which means it is a shorter circuit than the linear flow. During the cyclic flow, the electrons cycle back from the ferredoxin to the cytochrome complex and then continue the P700 in the PS1 reaction center. There is no production of NADPH and no release of oxygen, but this flow does generate ATP for the plant to use. Cyclic flow is considered an evolutionary leftover, but it allows plants who don't use this flow to grow in lower light and to not grow in intense light, which helps some of them survive when there is limited light. Oxidative phosphorylation in mitochondria and phosphorylation in chloroplasts have many similarities and differences which allow them to do their jobs in their specified areas. Both of these processes operate on the same principle, where they pass electrons down a chain of electron transfer agents in a series of redox reactions. As they are passed down, they pump hydrogen ions from an area of low concentration to an area of high concentration, which creates a proton gradient and allows the formation of ATP. The main reason these two reactions differ is due to where they occur. Photophosphorylation, which is the phosphorylation in chloroplasts, occurs in the thylakoid membrane of the chloroplast during the light-dependent stage of photosynthesis. During this stage, light in the form of photon supplies the energy needed to excite 2 electrons in the PS2, which are passed along a transport chain. Oxidative phosphorylation occurs in the membrane of the mitochondrial cristae during cellular respiration. The electrons are supplied by NAD and FAD, with oxygen acting as the last electron acceptor, leading the formation of H2O instead of NADPH, like in photophosphorylation.
The bullet that I had to focus on was to describe 2 important photosynthetic adaptations that minimize photorespiration. The 2 adaptations are known as C4 respiration and crassulacean acid metabolism, also known as CAM. C4 respiration is the adaptation of plants that starts with the 4-carbon sugar, which is unlike the normal Calvin cycle because that usually starts with the 6-carbon sugar. And the C4 plants were given their name because C4 means 4-carbon. The C4 plants attach carbon dioxide to phosphoenol pyruvate, which creates a 4-carbon compound. This process occurs in the mesophyll cells, which are the loosely packed cells in a C4 plant. Next, the 4-carbon sugar is pumped into another set of cells known as the bundle sheath cells, and these cells can be found in the vein in the leaf of the C4 plants. Then the carbon dioxide is released to be used by rubisco, which is used in the Calvin cycle to catalyze the first major steps in carbon fixation. C4 photosynthesis adaptation overall minimizes the photorespiration by spending ATP and by focusing on enhancing the production of sugar in the plant. This photosynthesis adaptation, you will come to find, is usually used in plants that keep their stomatas partially closed during the day because of the heat. The other type of photosynthesis adaptation I mentioned earlier was CAM. Just like the C4 plants, CAM plants also initially attach carbon dioxide to the phosphoenol pyruvate to create OAA, which is known as oxaloacetate. The differences between these two plants is that, unlike the C4 plant that fixes carbon during the day and pumps OAA to other cells, the CAM plant fixes carbon at night and stores the OAA in vacuoles in the cell. Doing this allows the CAM plants to open their stomatas at night during the cooling temperatures and avoid water loss. The plants then use the carbon dioxide during the day for the Calvin-Benson cycle so that it can be driven by the sunlight. The CAM plants that I've been mentioning are commonly known as cacti or a wide variety of succulent plants, and these plants are actually more common than the C4 plants. I think that C4 respiration and CAM both relate to big idea number 3, which is that living systems store, retrieve, transmit, and respond to information essential for life processes. I think this because during the C4 respiration process, the plants have learned to transmit carbon dioxide to phosphoenol pyruvate, which then creates a 4-carbon compound, and doing this kickstarts the process of the Calvin cycle and then leads to the first major steps in carbon fixation. I think that CAM plants also relate to big idea number 3, since they have learned to store OAA in the large vacuoles in the cell and to also fixate carbon at night because that's just what works best for these specific plants. Both C4 plants and CAM plants have learned the specific strategies and techniques that help each of them with their own life processes. C4 respiration and CAM both relate to big idea number 4, which is biological systems interact, and these systems and their interactions possess complex properties. Both C4 plants and CAM plants have certain step-by-step processes that are performed for specific outcomes that benefit the plants. Both C4 and CAM produce complex properties. C4 respiration produces sugar and ATP, which both minimize photorespiration, and CAM produces OAA, which is stored in the vacuole, and carbon, which is used in the Calvin-Benson cycle.
Describe the role of ATP and NADPH in the Calvin cycle. The Calvin cycle is an anabolic cycle that builds carbohydrates from smaller molecules and consumes energy. This cycle spends ATP as an energy source and consumes large amounts of NADPH as reducing power for adding high energy electrons that will make sugar. The Calvin cycle will then use the chemical energy from both ATP and NADPH to reduce the amount of CO2 in the sugar. The Calvin cycle incorporates each CO2 molecule by attaching it to a 5-carbon sugar called ribulose biphosphate. They join together to produce a 6-carbon intermediate, which is then short-lived because it is very energetically unstable, which causes it to split immediately, forming 2 molecules of 3-phosphoglycerate. Each molecule of the 3-phosphoglycerate receives 1 additional phosphate group from ATP, causing it to become a 1,3-biphosphoglycerate. The reduction phase will then begin with the reduction of 1,3-biphosphoglycerate by using the NADPH. During the process, it will lose a phosphate group, becoming a glyceraldehyde 3-phospho. There will be a total of 6 glyceraldehyde 3-phosphate molecules because there are 6 1,3-biphosphoglycerate and 6 NADPH molecules. 1 G3P molecule will exit the cycle and be used in the plant cell. The other 5 will be recycled and used to regenerate the CO2 acceptor ribulose biphosphate. 5 of the G3P molecules will be acted on by 3 more molecules of ATP, which will allow it to continue the cycle. Describe the major consequences of photorespiration. Photorespiration is the process in which occurs in light and consumes O2 while producing CO2. Unlike normal respiration, photorespiration uses ATP instead of producing it, and will not produce any sugar like photosynthesis. Photorespiration decreases photosynthetic output by siphoning the organic materials from the Calvin cycle and release CO2. The CO2 that is being used would have been fixed in the Calvin cycle, but will now be seen as revolutionary baggage. This is because it was more efficient when the Earth's atmosphere had more CO2 than it did O2. With such high amounts of oxygen now present in the Earth's atmosphere, the only positive aspect of photorespiration is that the light is able to limit the progress of the Calvin cycle. This is clear that the photorespiration acts to neutralize any damaging products of the light's reaction. If the carbon fixation was fixed and photorespiration was to be removed, crop production would be able to increase. This would occur because the Calvin cycle would be able to function more efficiently, allowing plants to grow at a faster rate. The roles of ATP and NADPH, along with photorespiration, correlate with big idea number 2, energy. ATP and NADPH are both energy-carrying molecules that are needed for light-dependent reactions. Without either molecules, energy would not be able to be produced. Although photorespiration does not produce energy, it does use the energy that is being produced during photosynthesis. This process uses the energy because it fixes carbon, which will be used for subsequent carbohydrate synthesis.